Okay, and we're going to get started now. So hello everyone, and thank you for joining us today. Welcome to today's Tips and Tricks webinar focusing on how to optimize your localization data clustering hosted by ONI. So before the webinar begins, we'd like to go over some quick house rules for the webinar. I'm sure you've all noticed by now, but you do have a webinar navigation panel with different sections. If you'd like to more clearly see one specific section, you can click the expand icon located in the top right corner of each section. Today's webinar will last about 45 minutes, leaving approximately 15 minutes for Q&A at the end. So you are welcome to submit any questions you have throughout the webinar via your chat section located within your panel. And we're going to be answering your questions during the Q&A session at the end. And if you're unable to get to these questions, then we are going to email your answers separately via the email address that you did register with. So within your panel section, you will also find a handout section. Here you can find our company brochure telling you a bit more about ONI and our offerings. So please also feel free to have a look there. And please note that today's webinar is being recorded. And if you'd like a copy of the webinar, please feel free to send us an email and we'll be more than happy to share the webinar recording with you. Now that we've gone over very quickly the house rules, I would like to introduce to you Haraman Johal. And Haraman is a data scientist at ONI focused on automation of experiments and analysis of super resolution data. Prior to joining ONI, just over 5 years ago, he studied for a master's in engineering science. And now I will pass the presentation over to you, Haraman, to begin the webinar.
Thanks, Carolyn. Okay, just want to confirm everyone can see my slides and everything. Then I will go ahead. So, thank you all for joining us. Excited to talk about clustering today. As Carolyn mentioned, I have worked on this area for a while at ONI, as well as lots of other things. So it's great to see that there's a lot of interest in discussing it. So today we're going to give you an overview of clustering and how it might be relevant to your data. We'll be talking about why clustering is important in the first place in this domain. We'll introduce you to a little bit of how clustering works. And then we'll go through a case study together on immune cells. We'll be looking at the clustering of receptors. At the end, you'll have some time for questions, as we've mentioned. So let's get started. Now, when I think about a scientist's day-to-day work, I think about this kind of valley of discovery. And that means that you're working through a series of steps to progress the scientific understanding of your topic. Now, the way I think about this is you start at your kind of biological, like high level, where you're asking some kind of life science question. You then need to understand the context around that question and design an experiment. And then when you're all the way down in the weeds, you're able to collect some raw data. And then you need to bring that raw data all the way back up to the level that you can interpret, which means you need to pre-process that data and you need to essentially interpret that data so that you can find the answer to the original question you had. So because we at ONI are in the business of super resolution microscopy, I'm just going to give a quick recap now on our type of data that we collect, which is localization data mostly. So if you imagine a labeled molecule inside your sample, generally what happens is we excite that labeled molecule with some kind of laser, and that molecule, as that label we're talking about, is a fluorophore. So that fluorophore kind of accepts this laser excitation and then gives off some kind of emission light. Then what happens in our microscope is that we capture that emission light using a camera image, which in this case might look something like this. And using that image, we can then fit a localization, which is essentially a point, to a sub-pixel accuracy. Now, obviously, this molecule is much smaller than a pixel, and the image that it produces is much larger than a pixel. But like I said, with that kind of Gaussian fitting, we're able to deduce where that molecule is to a greater accuracy within that pixel. So we call these points localizations, where one localization corresponds to that blinking of that labeled molecule. So now that we understand the raw data that we're working with, we can imagine that over a whole experiment, many molecules throughout time are blinking and creating localizations. That builds up into a full point cloud, which kind of is an alternative to an image. Instead of pixels, we have coordinates, and all of those points together form our data set. So let's now think about that same valley of discovery, but with localization data. In this case, once you've designed your experiment, you collect this raw localization data, you then process that data, and here is where you want to relate those localizations back to your biological question, and that's where you would need clustering. So that's this part here. We're clustering our data, and then we are analyzing those clusters in order to find the answer to our original question. So now that we understand where clustering fits inside the workflow of these experiments, let's have a look at how clustering works in general. So we're going to start with a quick poll. We're going to ask you how many clusters you see in this example point cloud that I put in front of you. Take a quick moment to have a look, because I think once the poll appears, it will cover the whole screen. And then I think, Carolyn, you're going to activate that. Cool. So the poll is now in progress. Just give you a couple of minutes or a couple of seconds, I should say, to answer that.
We have a few responses coming through, Haraman. I'm just going to give it another few seconds.
Yeah, no worries. All right, and I can see about 70% of you or so have voted, so let's maybe wrap it up there. And I can see the results here. Most people are saying there's probably 3 clusters. Some people have gone for some other values. But let's have a look now at something that might change that opinion, which is what if I gave you some more context, right? So you looked at that particular set of localizations and you said there are 3 clusters. But now if I introduce more data further away in similarly shaped groups of points, maybe you would change your answer to that same poll. And the point I'm trying to get across here is that clustering is by its own very nature like a hierarchical problem. It means that depending on the scale that you look at your data, you may see different types of clusters emerging. And some of the algorithms we're going to look at shortly will kind of account for that behavior. I think it's also healthy just to take a moment to think about how the human brain forms these clusters, right? We look at these points, how far away they are from each other, and we say locally, these points are roughly the same distance away from each other, so maybe that is a cluster. And all of the algorithms that people have developed in the clustering world are to deal with that kind of natural process and try to formalize it into some maths. Obviously, we want to use quantitative methods that are encoded in algorithms so that our data is not being affected by the bias of what we see as humans, but is kind of reliably and consistently analyzed in every case. So let's move on and have a look at those same points that we know and love, and think about how we might break these down into clusters. So what I'm going to do is say, on the left, I'm going to label the clusters of localizations, and on the right-hand side here, I'm going to introduce this concept called a cluster tree. Now, there are many ways to do clustering, but the methods we're talking about here in this webinar will mostly be using a cluster tree. So let's start to understand that concept. Now, the most naive thing we could do is to say all of these points are part of one big cluster. So that would be a ring like this. So it encompasses all these points, and we're going to introduce the root of our cluster tree on the right-hand side. Now, if we were to say, be a bit more stringent with what forms a cluster, and we say, okay, we're not going to allow things to be this far apart in our cluster. Let's maybe break it down a different way. We could say there's one cluster here on the left, and these 4 points on the right, they're a bit further away, so they're probably their own cluster. And you can see how these two pink and blue clusters are kind of children of this original white cluster. And that's how we break it down on the cluster tree as well. So you can see these 2 nodes are splitting off from the main root. And we could go even further. As many of you said, there do appear to be 3 clusters if you look at it in a certain way. So what we can do is break down that pink or magenta cluster on the left into this cyan and this purple. Now, even if we do that, we may not actually want to break down the blue cluster on the right-hand side. So we might want to kind of retain that. And what we're saying there is that blue cluster is already of some kind of minimum size of a cluster and doesn't really benefit in terms of like the analysis from breaking down even more. And using different types of metrics, the algorithm actually is able to decide when a cluster doesn't need to break down any further. Maybe it's reached some kind of limit. And you can see here the blue does not have any further children in the cluster tree. So this is one way in which we can kind of visualize the possibilities of all of the different clusters in a tree. And it's important to notice, though, that you can't have all of these clusters at the same time. It would be meaningless, for example, to have both the white cluster and this pink cluster, because then you're counting the same points here in two different clusters, and that will obviously not be appropriate for your analysis. So what you'll find is that if you take kind of a cut across the tree, which would be here like the pink and the blue, or a cut which kind of wanders across the tree, which is this cyan, this purple, and this blue, that would be a reasonable clustering result, because those are clusters that can coexist without counting the same points twice. Okay, so hopefully that's a clear enough explanation of how the cluster tree is formed. Let's now kind of look at a slightly bigger cluster tree to talk about the algorithms that we actually use in our analysis software. So our points go into the clustering algorithm. They kind of derive this cluster tree. We're not going to go into the maths of that in this webinar, but feel free to reach out and contact us if you want to know more about it. And then once we've got this cluster tree, there are many things we can do. Now, one thing to remember is that this kind of vertical axis in the tree is also meaningful. It means that at the top, we have very big clusters, which are very permissive. They allow very large gaps between the localizations in our cluster. And at the bottom of the tree, we have these kind of smaller clusters, which are very tight. They don't allow very long distances. So as you can look at this tree, you can think about small distances between points at the bottom and large distances at the top. Okay, so let's look at our first clustering method. This is dbScan. You may be familiar with this method already, as it's quite widely used in the life sciences. This is density-based spatial clustering of applications with noise. And this method is quite well established, and what it does is it effectively looks at this cluster tree and takes a horizontal slice across that tree. Now, that kind of distance value that it's got, I put in quotation marks here, is really like, where should this horizontal line be? Should it be at this point, or should it be higher up the tree or lower down? And that is really the main parameter for the dbScan algorithm. You're able to tune this value so that you can say, okay, I want clusters that are more dense or less dense by moving this line up and down. So once you've chosen this cut across the tree, those clusters that you've cut through are the ones that will be in your final result. Remember, the tree is just all the possible clusters, and we want a certain set of those, which really describes our data very well. Okay, so that's dbScan. It's really good, as you can tell, at getting clusters with similar properties, because it takes this horizontal cut. All of those clusters are going to be of a similar density, and they kind of have like a uniform, maybe size and like density of points. So that means that when you're in an application where you know the clusters are roughly the same size or roughly the same density, it's a good algorithm to use. Let's move on now to HDBScan. This is another published method. You can see the citation down here. And this is very similar to dbScan, but it's hierarchical. So it actually allows you to take a cut across the tree, which is not necessarily straight. So in dbScan, we said we want all the clusters from the same level because they have approximately the same density, and that's interesting to us. But in HDBScan, we allow the algorithm to use another metric, which is called stability. Now, I'm not going to go again into the maths or the details of that right now, but what you can think about with stability is that it's the likelihood that this cluster would break apart into children. So a cluster that's very easily separable into two might be a unstable cluster, but one cluster that you can't really break apart in a meaningful way would be less stable. So HDBScan computes the same tree, and it decides, okay, I think that these are the most stable clusters. So in some cases, it might pick out smaller clusters, in some cases larger. in some cases sparse clusters, in some cases more dense. And that's often useful in cases where you have clusters with variable density. dbScan will not allow you to pick out multiple populations of quite different clusters in the same data set, but HDBScan may be able to help with those cases. So because you don't have to enter a parameter to kind of select like you would with dbScan, it can be more straightforward in some cases to use HDBScan, but obviously you have to take that with some understanding that the algorithm is making choices, and those choices may or may not be appropriate depending on your application. Okay, so this is our second method, HDBScan. And here at ONI, we've also kind of expanded on that HDBScan method in something we like to call constrained clustering. So we take that same cluster tree, and you can see here I've colored some nodes because I want to use them for reference in a little bit of an explanation. And what we're going to do here is say we want to exclude clusters based on certain properties like area, maybe radius or skew. And I'll explain a little bit more about that in a second. But essentially what we're saying here is we have information, maybe from the biological context of our experiment, that tells us we want clusters of a certain type of shape. And then we can use some parameters to basically apply those pieces of information such that our clustering result is a bit more meaningful. So let's have a look at a concrete example here just to make that a bit more understandable. We're going to start with skew. So skew is a measure of kind of how stretched out is a cluster, how long and thin is it? And the way we do that is we take the length of a cluster along its longest dimension, and we divide that length by the width. Now, if you have a very like kind of round or square cluster, the length and the width are going to be pretty similar. So they're going to be around 1. But if your longest dimension is like 10 times as long as it is, the cluster is wide, then you're going to have a very long and thin cluster, and you can divide that 10 by that 1 to get a high skew, higher value. So this is something we can do. If we get the cluster tree on the right-hand side, then for every cluster, we can say, calculate this metric, skew. So let's maybe have a look at these green points I've left in the bottom left and think about how that would apply. So I'm going to say that this pink cluster, which includes all of these points, corresponds to this pink node over here on the tree. And as you can see, this shape that the cluster makes is quite long and thin. So we might label that cluster as having a high skew. I'm not going to actually calculate numbers in this case, but you can see by the shape of the cluster that it would respond to this metric being quite high. Now we're going to follow the same process we did earlier, where we break this cluster down further. And let's say the next nodes in the tree are this purple and blue node. So this purple corresponds to these points here, and this blue corresponds to these points in a cluster bottom. Now these two clusters are a bit more round, right? They're not long and stretched out. They're roughly square or circular in proportion, so their lengths and widths are going to be very similar. So they won't necessarily have a high skew. But if we go even further and we break down this blue cluster into two smaller clusters, you can see that that cyan at the bottom and that magenta color, those are both quite long, thin clusters, and therefore would also possibly have a high skew when we measure that metric. So now we have more information about our clusters. We can actually use that information to improve our clustering result. Let's say we're in an application where we expect to have kind of round clusters and we don't expect to see anything long and thin. Then we can apply a constraint on skew so that we can remove those possible candidates from our cluster tree. And if we move forward here, you can see I've crossed out those 4 nodes, and that might affect which clusters are then selected in the stability kind of assessment that HDBScan normally does. So in this way, you end up with these 2 clusters, which are the ones you wanted, because you know that your cluster should be round with a low skew. And you don't have to throw this data out, and that's a very important point. So when you know the cluster properties in advance from your context, you're essentially able to say, I'm going to constrain and then pick out these clusters. But importantly, if you just filtered here and you were selecting, say, these 2 clusters at the bottom by HDBScan, as we saw earlier, those clusters would just have a skew that was not compatible with your filters, and you would simply throw out this entire part of the tree. you wouldn't be able to analyze those clusters at all. But the advantage of constraints is that it allows you to keep that data by picking a cluster that does match your properties, but is still, is still stable enough to be a reasonable choice. Now, this is quite a lot to take in, and it's relatively detailed, but we're going to go through a live example shortly where this should become a bit more clear. The important thing is that when we constrain, we select different clusters rather than just simply throwing out clusters that don't match our requirements. Okay, so we've been through 3 different clustering methods, so let's have a quick recap of what those were. We had dbScan, which is a very familiar method for finding clusters with a kind of uniform density and similar properties. Then we had hdbScan, which is another published method, which is an extension to dbScan and allows you to pick out clusters with different types of density. And we also had constrained clustering. And in this case, you can apply more information about the shape and morphology of those clusters, which you may have from the biological context. Okay, so these are the 3 methods, and we're going to have a quick look at how those are used in practice in our case study. So first, we're going to go through some context, then I'll show you a live example where we perform clustering on our analysis platform, and then we'll have a look at those clusters in analysis and see how we might be able to interpret the clusters that we have found. So just a little bit of context to start with. As Carolyn's introduction suggested, I am not a biologist, so this is a little bit outside my comfort zone. But from my understanding, we're looking at immune cells, and we're looking at specifically receptors, which are these grey vertical structures poking through the cell membrane. And we're going to be looking at how, when they go through a clustering process, we should be able to detect the clustering of these receptors, and that may be interesting to us from the point of view of a biological question. So just a little bit of context, we have on the left-hand side some receptors which are maybe spread out throughout the cell membrane, and there are these effectors and adapters which are kind of swimming around inside the cell, and they may bind and unbind to the receptors on the cell membrane. Now, when the receptors are kind of dispersed across the cell, this binding and unbinding happens at a kind of low rate, which is not particularly of note to the cell, should we say. But as the receptors are clustered together, perhaps by an internal active process or by an external force pushing these receptors together, we actually end up in a cascade situation. Now, because there are effectors and adapters binding and unbinding from the receptors, in that vicinity where there are lots of receptors clustered together, this binding and unbinding happens at a faster rate because there are more candidates for binding and there are more binding sites available. And this can lead to a kind of runaway cascade, and that means that that signaling may trigger some response from the cell or may cause these receptors to kind of clump even more tightly depending on the mechanism. So this is an interesting process that we might want to look at by labeling these receptors and seeing how they cluster in different conditions. So we can expect, if we're looking at a cell here, which is bordered in white, in a non-signaling case, we should see quite dispersed receptors, which you can imagine we've labeled with this kind of green fluorophore. And in a signaling case, like on the right-hand side, we should see these receptors clumping together into tight clusters across the cell. So this is what our context is. And today we'll be looking at a specific question, which is, using a couple of different capture surfaces, how do the surfaces themselves affect the receptor clustering? So we're going to be looking at a capture antibody case, and we're going to be comparing it to a PDL. And the interesting things to keep note of is that the PDL is known to partially activate the surface, and the capture antibody, in this case, captures the T cells and sticks them to the surface, but it does not activate the cells in a meaningful way. And there's a citation there on some related papers about how we come to know this. But essentially, we're going to be looking at the same cells in 2 different situations on these 2 surfaces and trying to decide whether the clustering lines up with our expectation about the activation. Okay, here's a quick note on the experimental protocol. If you have any questions about this, please do let us know and we can get back to you after the webinar. As I said, this is not my area of expertise, but just for context, this is the kind of workflow we're going through with these cells. And now I'm going to move to the live example, which is clustering on Cody. Now, Cody, you may or may not have heard about before. It's ONI's collaborative discovery platform. And this is a place where you can upload your data, you can store it there, you can organize it into different types of collaborations, and then you can obviously analyze that data, you can visualize it. This is all happening in the browser, and it makes it very convenient for you to access your data from any machine and also to share it with your colleagues if you need to, or your collaborators. Any analysis you do on one data set can then be applied across a batch of similar data sets. And this kind of workflow really, we hope, will speed up your experimentation and enable you to take your localization data analysis to another level. So now I'm going to switch over to that software and we can take a look at how this clustering works in practice. So that should be over here. All right. So we're immediately presented with a dataset view. You can see that these are the localizations I was talking about earlier. These individual points are those localizations. And you can start to see possibly some tight clusters in this data too. But let's zoom out and get some context first. This is one cell, and you can see here the dataset title. We're in the PDL case. And if we zoom out even further, you can see the whole field of view. So there are various cells in this dataset, but today, for simplicity, we'll just be looking at this one. Now, what else can we see in our view right now? We can also see this region of interest that I've drawn. Now, Cody allows you to apply these regions of interest, and we're doing one here because at the edge of the cell, we have other types of effects, like the membrane folding over on itself, and that might affect our clustering results. So we don't want to be kind of biased by strange things happening at the boundary. So we'll be looking at this contact surface where the cell is cleanly just against the glass. So this is one way to restrict your analysis to a certain area. Now, what else is going on the screen? We've also got some visualization settings on the right-hand side. We've got different layers in the visualization, like the region of interest here. I can turn it on or off. We've got some clusters that we'll be looking at shortly. And we've got some various display parameters that we can change. In the top right, you can see that I'm logged into the Cody platform, and I'm able to search Cody for different types of data sets, different types of experiments. And on the left-hand side, this area is regarding analysis. So you can see we've got one analysis widget already open here. This is the clustering widget, and you'll see how that works shortly. But if we look at this workflow on the left-hand side, that's also quite interesting. This is for a kind of single molecule workflow, and you can see here we've already got some steps in green, which means that they've already been run. So I've got a drift correction that I've already applied to this data set. I've applied some filters, and I've drawn this ROI, this region of interest, in the annotations tool. So all of these steps have been done before the webinar because today we're focusing on this tool, which is clustering, which is the one in progress. And obviously that pre-processing is important, and clustering is just one step in that valley of discovery. Coming out after the clusters, we'll return to the slides and see how we might interpret this data. So let's get started now looking into the clusters themselves. In this cell, we've analyzed this area inside the region of interest, and I'm going to pull up the clusters here. So what do we see first? A very large pink cluster. In this case, the colors of the points are going to tell you which cluster they are in. So you can see one big cluster here, and this lines up with our expectations because currently we are in dbScan, and we have a very high EPS value. And this distance parameter is telling us we're going to permit very large distances between points, and that means that we're going to end up with almost all the points in one giant cluster. Okay, so what happens when we adjust this parameter? So you can see as I reduce this distance parameter, our big cluster starts to break down. And one nice thing about our collaborative discovery platform is that we can respond in real time with these parameters. Some of the computation is happening on the cloud, but some of it is also happening locally so that you can very quickly adjust your parameters and get some immediate feedback on what's going on in your data. So you can see here, we've got these very large clusters. But if we keep going down, the clusters will get smaller. Now let's remind ourselves that the clusters we're actually looking for are the ones in this data set. And if we were to look by eye, we can see that these clusters are relatively tight and small, kind of like this guy here. And those are the ones we really want to pick out with our analysis. So let's reduce that EPS value even further and see if we get down to around, I think it's 30, you can start to see very small tight clusters like the ones we're looking for. Now this is quite nice, and being able to toggle on and off the layers here shows you that we're picking out clusters that are reasonable. This cluster here we can see by eye, and then it's selected by the clustering algorithm too. Now, obviously in a more in-depth experiment, you wouldn't want to just slide the slider until the clusters look right. That's not a very unbiased way to apply your parameters. In reality, what you'd want to do is take a negative control of some kind. In this case, you could imagine putting the same labels into the cell, but without binding them to the receptors. In this way, you should end up with some kind of uniform background throughout the cell where these labels are just floating around. And they're probably not clustering in any meaningful way because that's how it's a negative control and they're not going to bind. So then if you apply some parameters to a positive control, like this case, and the negative control, you should find that the same parameters get quite strongly different results. In the positive case, you'd expect to find the clusters like this. And in the negative control case, you probably wouldn't find almost any clusters at the same parameter level. So that's just one example of how you might go about setting these parameters. But in this case, we're just looking at this one data set today. So we can see that we've picked out a bunch of clusters here with dbScan. And if we wanted to know more about them, we can actually save these results. As I mentioned, when we're changing the slider, we're changing in real time just on our own computer. But by saving the results here, we're actually running a process on the cloud so that those results will be available to anyone else who wants to look at them, who you've shared this data set with. So at this point, you can see we've reloaded back in that data, but now we have more metrics. So we can see the area distribution of these clusters, which is some kind of Gaussian here, you can see, and maybe other properties like the radius or the densities of those points, of those clusters, sorry. So this is a little bit of the capabilities of Cody, and all of these statistics here are like kind of indicative of this distribution. And you can kind of click around too to look at the clusters and see, okay, this particular cluster has 15 localizations. This is its area, this is its density. And as we step through skew, for example, it will show up these different properties underneath that kind of label. So this is a nice interactive feature of our platform, which allows you to kind of look both at the graphs and at the data in a natural way that you might be able to investigate things. So this is how dbScan works. We're able to set that one parameter, which, as you'll remember, is sliding that cut up and down the tree. And now we're going to take a look at some other clustering methods that we've talked about. So I'm going to load now an HDBScan result, and we'll see how it's different to this clustering. Now, you may have just missed that, but previously in the dbScan result, we had 670 clusters. Now, that number is just a bit of an indication of how much of the data is being clustered. And you can see here, we also have the clustered localizations, which tells you how many of my localizations are making it into clusters. You can see we have a larger number of clusters in HDBScan. And that's partly because it's kind of intelligently choosing that cut, but also because it's probably clustering some things that we might not have done by eye. Now you can see over here, we've got a more sparse cluster, and you can also see maybe on this side, we've got this kind of long stretched out pink cluster. And as we discussed earlier, like not all of these clusters are biologically relevant. Like if we were to look into this pink cluster a bit more, we might say, actually, there's either 2 or 3 clusters here rather than just one. And if we looked at the raw data just to make sure like that possibly is the way that we would do this if we were just annotating. But HDBScan does have the flexibility because of our constraint method on top of it to make those adjustments. So we'll get to that in just a second. But you'll remember that, you know, in the same way as dbScan, with HDBScan, we can get the same properties. We can look at the distributions of area, of radius, and then we'll be able to export all of that data later for our analysis. So this is what the algorithm HDBScan comes up with by itself. But let's say we want to apply some constraints. looking back at our data, we're pretty confident that actually we have these small tight clusters, and that's also what we expect from our understanding of the biological context. These receptors on this PDL surface are probably somewhat activated and therefore should be bunching together. And the clusters that we see do kind of line up with our expectations. So can we do something about the HDB scan result with constraints to pick out slightly better results? Now, just a brief reminder that if we just want to apply a filter, let's say we just wanted to filter out all these clusters with, like this guy, a high skew. You can see it has a skew of 3.1. That's the longest length divided by the width gives a ratio of 3. If we were to apply a filter which said skew must be lower than 3.1, we would just lose all of those points from our analysis. It would just delete this cluster and it wouldn't make it through to the next parts of our experiment. But if we look at this good cluster over here, it has a skew of around 1.2. So we might be able to set a constraint kind of somewhere between 1 and 3. Let's slide this slider down and see what happens. Now, this seems like a somewhat unexpected result. We've suddenly got these very large clusters across the data set. For example, this one over here, this red, is taking up a very large space. And that's because, remember, our HDB scan algorithm selects the most stable clusters. And after we apply constraints, we've removed some of those possibilities from the tree. And now HDB scan may find a completely different level at which to make its cut. So let's have a look now at how we can kind of bring that back into the range that we're looking for. Let's use the area slider to bring that maximum area down, let's say down to here. And you can see that we've now broken up those clusters again. And this real-time investigation with the parameters is very useful to be able to see what things are happening in straight away and then kind of respond to them. And again, if we look into that same cluster as before, you can see there are now actually 2 clusters here, which is definitely an improvement on that one big one, and also tells us that we were actually able to use some of that data that previously we would just be throwing out with filtering. So this is the kind of operation you're able to do using the constrained clustering. And here you can see, we've got more clusters than we did with dbScan. And that's because dbScan is not able to pick out these different clusters at different levels of the tree in the same way that HDB-scan and combined with constraints can do. Okay, so there's a lot there, but using this type of analysis, we can pick out clustering with dbScan, HDB-scan, and with constraints. And using this type of analysis, which I've done offline before the webinar, we can now revisit the slides and see how that data can be interpreted. Now, just so that you guys understand what's going on, you can see that if we save the results here, we're able to actually export that. And again, we're just running some operations in the cloud to save these results down for future. Then I can access over here, once that's completed. Yeah, you can see that we can download the clusters CSV, and that would allow us to do some more offline analysis. Now, as I mentioned, Cody is a platform with lots of different analysis tools. And right now, you can already do some types of counting on these clusters, and you can talk to us about that offline if you're interested in what's already available. And in future, we'll be adding many more cluster analysis tools, even to the UI, so that you'll be able to build up a visual analysis of your data set. But for now, let's have a look at how we can use that information about the clusters that we found and see how that affects our initial biological question. So I'm going to take us back to the slides now. And you can see here that we are moving on to the cluster analysis part. So let's assume that I've set those parameters well and I've done my clustering. How do I now relate those clusters back to my biological question? If we take our original data that we had here, just a reminder that we're looking at that cell in cyan, that was the PDL case. And I've got another cell over here from the capture antibody case. Now we can see by eye there are maybe some subtle differences between these two data sets, but we'll go into some quantification now and how we can kind of demonstrate that with some more robust analysis. So let's first think about HDB-scan. If we apply HDB-scan, it's selecting the clusters based on what it thinks are the most stable. Now let's compare what HDB-scan picks out for the PDL case and for the capture antibody case. So what I've done on the right-hand side is plotted all the clusters from each of the two cases on a histogram where we're measuring the density of those clusters. So in the cyan from the PDL, you can see a kind of stretched out distribution here moving over to the right-hand side of the graph. And what we're saying is that a high frequency of clusters have a higher density. Whereas in the capture antibody case, we see this magenta line, which has a higher frequency of clusters having a low density. And what we're saying here is that when you use PDL, your clusters in general are more dense. So this lines up with our expectation that PDL should somewhat activate the surface, should somewhat activate the cell and therefore cluster these receptors. So this is one way in which we can interpret the data. And then another way we might want to look at it is using the metrics of other things in our analysis. So because we have drawn our region of interest in both cases on Cody, we have access to that area of that region of interest, and then we can kind of measure how many clusters we find per unit area of the cell. So if we simply divide the number of clusters by the area of the region of interest, we can get kind of a density of clusters, not of localizations in this case, the density of clusters across the surface of the cell. And in that case, we also see more clusters in the PDL case compared to the antibody case for the same unit area. And all of these metrics are essentially pointing to this idea that the clusters that we find in this data point to PDL cell being more activated with tighter clustered receptors than in the capture antibody case, which should not affect the receptor clustering. So this is using HDV scan, and that was kind of a default selection of clusters from this algorithm. But we can also go about this in a slightly different way, just to show you the power of the other clustering algorithms. We could look at DB scan. So remember, with DB scan and a low EPS value, we can start to pick out the right clusters from the PDL case. And you can see lots of small clusters here. And you can see kind of how many clusters are found in the cell. Now, if we apply that same parameter, so the same analysis to the capture antibody case, we find far fewer clusters. We can see the difference in the number of clusters here, and also how many of the localizations even make it into a cluster. So you can see in the capture antibody case, when we're doing this DB scan analysis, we're actually throwing out a lot of the localizations and not even clustering them at all. And that's because essentially with this DB scan value, we're saying we're looking for clusters of a certain density. And the reality of the data in the capture antibody case is that there are not that many clusters which actually meet that criteria. So here we're showing that this tight cluster is really much more prevalent in the PDL case, aligning with our hypothesis. And finally, we're going to show the similar analysis, but this time using constrained clustering. Here we've applied some constraints on skew, area, number of localizations, and radius. All of this is based on the idea that we know what these clusters look like in advance on these clusters of receptors from our context. And we can pick out many of those clusters on the left-hand side. And in this case, because we've so clearly described the morphology of these clusters, because of what we know about the sample, we get an even starker difference when we apply the same constrained clustering to the capture antibody case. So even less of the clusters are picked out in this case because almost nowhere in the cell do the localizations meet this kind of stringent criteria. So these are a few different ways in which we can demonstrate using the clustering methods, metrics and quantifications, so that we can interpret these clusters into a level which allows us to relate them to the biological question. And this is how you might go through an experiment, obviously with more negative controls and maybe more repeats than just one cell. But for today's example, hopefully you understand that with this toy example, we can use these different clustering methods for different types of analysis. So now we're just going to have a quick look at a couple of other clustering applications and how this analysis technique might be useful in the wider world of the life sciences, and then we'll have some questions. So we'll have a quick look here at the case of tumor metabolics. So in the left image, you can see the raw data. And what we're looking at here are nuclei stained with lamin in cyan and some mitochondria stained with TOM20 in magenta. Now, in this experiment, we might be interested in how these mitochondria are distributed, where they are, maybe even in relation to the nucleus, or maybe about their shape and their size. All of this information could be interesting. And you can see on the right-hand side that using clustering, we can cluster that magenta channel and then pick out these mitochondria. And you can see kind of a detailed view here of this section. And when we have the mitochondria clustered like this, we're able to then perform some kind of quantifications on the shape of the mitochondria and maybe their dispersion across the sample. So this is one way in which you might use clustering differently. And if you're familiar with ONI, you may have heard that we're quite keen on extracellular vesicles. In this case, you can see maybe in the bottom left image, we've got like some raw DSTORM data. And then if we were to cluster that data, we can find kind of each EV with one cluster, assuming that we're able to set our parameters correctly. And you can see here, we get quite nice separation of each EV corresponds to one cluster. And if we have some kind of surface stain, which is kind of reliably getting every EV, then we can then use those clusters to then decide where to count maybe surface markers that we're interested in, sorry. For example, like CD9, CD81, and those kind of things. So we can pick out the clusters, and we can therefore pick out the EVs, and then we can perform further analysis on each EV as kind of a single object by looking at other channels of information. Okay, so with that, we are now pretty much all the way through our webinar main content, and I'd like to welcome you guys to ask any questions about single molecule applications and clustering. We operate in many different fields of life sciences, and this type of analysis is very useful to many different domains. So we'd love to hear from you about that. And I think I'll hand over now to Carolyn, who's going to maybe just have a little spiel here at the end.
Great. Thank you very much for that presentation, Haraman. I am hoping and I'm sure that everyone learned some new clustering techniques today. We are now going to open up the floor to questions for Haraman, which he will be answering live right now. And once again, if you have any questions, please feel free to drop them into the questions or chat box. It looks like we already have a bunch submitted, Haraman, so we are going to start with the questions. First question we have is, how can I know if I should choose HDB scan or DB scan? Could my results be wrong depending on what I choose?
Right, so that's a good question. When you're choosing any clustering method, and I'm sure many people in the audience have used things that are not even in the options that I talked about today, you should have some like working level of understanding of the method and then also of your biological context. And as I said earlier, like depending on the context, you may know more or less information about your clusters. And I'd recommend if you're in a case where you don't really know what kind of clustering the biology would imply, then you should kind of enter with an open mind and try a bunch of things first. We'd be very happy to also talk to you about that process and figuring out which method is appropriate. But in broad strokes, HDB scan should be able to pick out different populations of clusters, and DB scan should be able to pick out kind of more uniform populations of clusters where you're expecting clusters to have similar properties. Hopefully that answers that question.
Great. Okay, the next question we have is, what if I want to use a different algorithm that's not on ONI's platform?
Yeah, so this is a great question. And one thing that we're building with the collaborative discovery platform that I introduced briefly today is a space for any kind of analysis algorithm. And that means that we'll be adding more and more methods to this platform. We already have DB scan, HDB scan, and this constraints method that we've kind of developed. But we would love to add other methods like Voronoi clustering or any other kind of k-means or things that you're interested in using. And one thing that's very exciting about Cody is that in the future, we're hoping that you scientists will be able to contribute your own methods to the platform. So that means you'll be able to add your own methods of analysis and your own kind of steps here, not just for you to use, but even potentially for you to share with other people and kind of increase reproducibility across the life sciences in that analysis space.
All right, wonderful. The next question we have is there a specific order that one should follow when applying the constraint?
Yeah, so this is a good question as well. So those constraints can be kind of a new concept, and it might be a bit confusing how they work, but the way I'd approach it is kind of just look at the first result from HDB scan, and then think about what is it about the clusters that you see that doesn't seem right. Now, you'll probably be able to describe that in words to another human being, but then look for the mathematical properties of the clusters that we've made available on the platform, and decide, like, does the idea that I have, like this cluster is not circular enough, or the cluster is too long and thin when I'm expecting something different, that probably has some kind of interpretation or some metric which we can then apply constraints to, and that's what you should be able to find on the platform. Now, when you apply these constraints, you should start with that one which you picked out, and then you might find, like we did today, that when you change like the skew, you end up with clusters of a different area, and then you may need to apply other constraints in order to bring us back down to the level we're expecting. But that's quite a natural process, and if you're worried about over defining the clusters that you're looking at, repetition is your friend across different cells, across different fields of view, across different samples, and you'll quickly find that any overtuned parameters will kind of break down in the general case. So you'll be able to find a natural set, hopefully, that you can apply more broadly.
Thank you for that, Haraman. Our next question is, don't you run the risk of counting the same localizations twice with constrained clustering?
So actually, no. The way that constrained clustering works is that it has this same principle as HDB scan, which is that you cannot have a cluster and its parent. So any clusters that are selected by the constrained clustering will be kind of exclusively using points. So no point can appear in two clusters by definition in HDB scan. And we keep that same logic to protect us from that same worry in constrained clustering as well. There is some checking in the algorithm to say, if this cluster is in, then none of these other clusters which are associated can be in.
Okay, our next question is, since a single molecule blinks multiple times, could you set a threshold for having a single molecule there versus multiple molecules?
This is a really interesting question. So we didn't talk about like how we process localizations today, but it is actually quite an interesting topic, and there are a lot of different considerations to keep in mind. When a molecule blinks, as you say, like it may blink more than once, and there are other interesting factors like the blink may last more than one camera frame, so it may be counted as two localizations. So actually, it's a relatively complicated problem, and setting a single threshold may not be possible naively on localizations. However, depending on how you process your localizations, like what kind of steps you do to filter, what kind of steps you do in sample preparation, and how your experiment is set up, like with is it a D-storm experiment, is it a palm experiment, like which type of localization microscopy are you using? There are ways to get around that, and we would love to talk to you about the details of trying to set thresholds and figure out quantitative ways to answer your biological question. But there's not a clear-cut way for me to say yes or no to that question right now, unfortunately. It depends, as I'm sure you're familiar with in many things in the life sciences.
Thank you for that, Haraman. Our next question is, can you only apply the constraints in combination with HDB scan or also with DB scan? Yeah, so
the way that DB scan works is it just takes kind of a horizontal cut across the tree. And what that means is you can only have a certain set of clusters. Now, if you wanted to apply constraints, what would happen is effectively you would just filter out those clusters. Because as the line goes across the tree, if you've constrained out a certain cluster, it just won't appear and it will be ignored by DB scan and you'll carry on. Now, what that means is that you're effectively just doing filtering. And that's okay, that's also an interesting operation, but it's good to have these two things separate in your mind so you don't get kind of down the wrong rabbit hole. But essentially, with HDB scan, you can apply constraints to get clusters, and then you can filter it. But with DB scan, the only option really available is after you've done your DB scan, you can filter the clusters. And that is also possible on our platform. So if you want to do that, you say, okay, I've done my DB scan, and now I want to like get rid of certain clusters that don't meet my criteria. That is also okay. It's just a filtering operation, not a constraining operation.
Thank you for that, Haraman. Next question is, what is the level of privacy for my data when I use Cody?
Yeah, so our vision for Cody is that it will meet the needs that you set out for it. And the way that you upload your data, you get to decide, do I want this to be a private data set, which only I can look at? Do I want it to be a public data set, which everyone on the platform can see? Or do I want to share this with certain collaborators? So you can essentially add your members of your lab or collaborators around the world to a certain collaboration project. And then they will all have access to the same data sets, but no one outside of that group will. So you have the kind of access controls that you might expect to be able to make use of the idea of like instant sharing across the globe with, you know, analysis in your browser, but you don't have the drawbacks of like, no, everyone can see this data suddenly. That only happens if you want to share that data. And it's actually something we would encourage. We would love to see more public data on Cody, as I'm sure would really benefit the life sciences, but it's not something we're going to force you to do. It's something that you would opt into.
Great. Next question is, what are the units of the distance tool? And Haraman, it looks like they asked this question when you were doing the DB scan clustering.
Right, so the units of that distance tool are not trivial to probably explain right now. I would say that there is a lot of literature about DB scan and HDB scan on the internet which explains this, and I'd be happy to discuss it offline in more detail. But essentially, they are taking a metric which you can think of as scaling with just spatial distance, but they're doing something a bit more complicated by using something called mutual reachability. And I'm not going to go into the details of that, but I'd be happy to explain it offline. You can think of a bigger distance at one end of the EPS scale and a smaller distance at the other, but the specific number of that value is not immediately interpretable. And that's one of the issues with DB scan. It's one of the things that different methods try to trade off on.
Okay, next question. It looks like they're just looking for a verification on something. HDB scan is not sensitive to sample volume, total detected links, in the form of a question.
Sorry, could you repeat that?
Yeah, they said HDB scan is not sensitive to sample volume.
So as you increase kind of the number of blinks that go through an experiment and the number of localizations that pop up, you will obviously end up with more points in your analysis. And this means that your clusters will be larger in terms of the number of localizations. They may also, if the same molecule is blinking more than once, you might see a small distribution around a certain point where that molecule is blinking. Obviously, this will affect your clustering because if you have constraints, let's say, on number of localizations or on area or things like that, then a longer experiment with more volume of points more volume of localizations, I should say, will end up with potentially different results. However, with HDB scan, it's still automatically, there are no parameters in that sense at least. So it will still try to pick out the clusters that make the most sense according to its stability metric. So let's say you ran an experiment and you ran it for like 100 frames. HDB scan would pick out some clusters from there. And then you ran the same experiment but for 1,000 frames. The clusters should have more points in them, but HDB scan should still pick out probably similar clusters because relative to each other, they will then like hopefully have all scaled in the same direction. And it might be that you find that in longer experiments, the way that the points distribute, you can like separate out artifacts that way. Maybe like if there is a piece of dirt in your sample, it may have different kind of blinking properties throughout time. And that will be something you can use to filter out different artifacts from your clustering. But yeah, if I didn't answer your question, I wasn't completely clear on it, and then feel free to follow up with us afterwards. And I'd be happy to add more detail.
Great, thanks, Haraman. I am going to give you one more question because we are almost out of time. There were a lot of questions that came through though, so I will tell everyone how we will answer them afterwards. But for the last question, can you analyze clusters within clusters? As in, can you run 2 clustering algorithms at the same time to detect how many small clusters make up a larger cluster?
That is a really interesting question. Yes, so there are a couple ways you can do this. The Cody platform is not yet able to do this, and I say yet because it's literally the kind of thing that we're working on right now, being able to do this kind of like tiered analysis or kind of chaining different tools. That's a very exciting area of development at ONI. But there are ways to do this. You could perform, for example, a dbScan clustering with a very like high X value to pick out like large clusters, let's say like a nucleus. And then once you've picked out those clusters, you could then apply another clustering with a region of interest which is defined by the first cluster, so that you're clustering only inside that nucleus, and you could do this for every nucleus. And then maybe you're picking out like nuclear pores, for example, and then those clusters would correspond to a nuclear pore. And we hope to be able to relate all of these levels back to each other so that you can say, okay, there were this many nuclear pore clusters inside these nuclei, and then across all the nuclei, I see this kind of distribution, or maybe these differences, depending on my conditions. So you definitely can do this. It's a little tricky with what we have set up right now, but we're very excited to be offering this type of functionality soon, and we'd love to talk to you if you have a case study where you want to do this. We'd be very excited to work with you. I should note one thing is possible already, which is that if you have two different channels, let's say you've labeled two different objects of interest, you can cluster those channels either as a group or separately. And that means that, let's say I have a cell with two different objects inside which are labeled differently. I can cluster those two objects independently, and then I'll get different metrics for each group of clusters to get whatever analysis I'm looking for exported.
Okay, and it seems as if we are now at time. So a very big thank you for the presentation today, Haraman. If we were unable to get to your questions, I know there's probably still over maybe 15 that we needed to get to. We are going to do our absolute best to contact you and answer your questions individually. And once again, this webinar was recorded. Therefore, if you need to refer back to it, you're more than welcome to do so. So thank you for all the attendees for joining us today. Upon exiting the webinar, you're going to receive a survey. You could fill out that survey to provide us feedback on your experience today, or tell us if you're in need of being contacted directly by ONI. That would be appreciated during that time. So thank you again, and I hope everyone enjoys the rest of their day.
Thanks very much.